Hi everyone, this is a video just to go through paper 1, January 2020. The idea is that I will do this under exam conditions and within the 2-hour time limit, hopefully to give you the idea, or at least a sort of flavour of how you should now, after you've done Easter revision, be approaching the exam in real time. If there's any teaching points, I will stop and elaborate on them if I think that they are required. Otherwise, I'll do this under time conditions and hopefully come in under the 2-hour time limit, both for your sake and for mine. So first question, diagram shows a food web. How many producers are there shown in the food web? Well, producers are photosynthetic organisms. So we've got producers here for the grass, the red oak, and the cranberry bush. So they are your producers. So the answer is 3. Talk about a mark a minute. How many secondary consumers are shown in the food web? Well, we go from the producers to primary consumers in blue, which will be the mouse, the deer, and the caterpillar. So the secondary consumers will be consuming the primary consumers. So this will be the tick, the coyote, blue jay, and the waxwing. So the answer is 4. So answer C. Uses some of the senses to catch deer. Name the sense that uses receptor cells in the retina. Okay, this is not asking for the receptor cells. This is asking for the sense that those cells in the retina detect. So the receptor cells, of course, are rods and cone cells in the retina. They detect light, and of course, the sense is therefore eyesight or sight. Let's go eyesight. A coyote runs 530 meters in 30 seconds trying to catch a deer. Fine. Calculate the speed in meters per second. That's quite easy because we're already in meters, and we're needing to go from 30 seconds to essentially 1 second. So we divide by 30. So all I need to do is do 530 divided by 30. And just to make sure I don't make any calculation errors, I'm going to, should have got my calculator already. What have I done with it? There we go. Whoops. Right, 530 divided by 30 is 17.66667. It doesn't tell me to do it in any particular sort of significant figures or not. So I think I'm just going to err on the side of caution and do 3 sig fig. So that is 17.7. So there we go. So the coyote will not catch the deer if too much lactic acid, okay, so I'm going to highlight that, builds up in its muscles. Explain why the lactic acid builds up when it runs. Okay, so we need to link running to lactic acid buildup in muscles. So this is absolutely a question about anaerobic respiration. So I would say the coyote begins to respire, or the muscle cells to respire anaerobically, producing lactic acid. And of course, this is in order, again, this is my explanation. Why is it even doing this? It's in order to produce enough ATP. And again, why is it important when it runs? For muscle contraction. To still occur in absence of oxygen. Okay. Happy with that for 2 marks. So moving on. Again, that probably only took about a minute, so we're doing quite well. Organisms can be classified into groups based on their features. State 3 differences. Okay, so I need differences between eukaryotes and prokaryotes in terms of organisms. Right. Okay, the first one, I think the most obvious one, is to say that eukaryotes have a nucleus. Oops. Have a membrane bound, that's important. Nucleus. Prokaryotes technically do have a nucleus, it's just not membrane bound, so we call it a nucleoid region or a nucleoid. The most obvious one as well is that eukaryotes have what we call organelles, so things like mitochondria, etc. So eukaryotes have organelles, e.g. mitochondria. There we go. And the third one, of course, is I think, again, quite obvious, the genetic material. So prokaryotes, e.g. bacteria, have circular genomes, circular chromosomes, whereas, of course, eukaryotes have linear chromosomes. Give an example of a disease caused by a protoctist. So these are the protists. They are eukaryotes, but they are not. They don't form within the kingdom of either animals, plants, or fungi. So they're kind of defined by exclusion, if you remember. And the sort of key one that you need to know is called Plasmodium. And it's Plasmodium falciparum, but I'm pretty sure you don't need to know the species name, but certainly the genus is Plasmodium. And the name of the disease that causes is malaria. Just of note, you can also have trypanosomes. So trypanosomes can cause African sleeping sickness, along with other things as well. But worth perhaps remembering the second one. But Plasmodium and malaria is the key one. But yeah, right, moving on. So we've at the end of question 2 there, so we got 5 marks there in probably about a minute or two. Squirrels are animals that live in woodland. They feed on nuts produced by the trees, right? Okay, so they eat nuts, fine. I don't really think we need a diagram of a squirrel eating a nut, but there we go. Describe a method that we could use to show that the nut contains fat. Okay, in other words, this is the test for lipids. But the key thing is that the nut is solid here. It's not a sort of aqueous solution or anything. So the first thing we're going to have to do is sort of grind it up. So step 1 is grind up the nut. And then typically we will add ethanol. You could grind it up in ethanol here, actually. You then add ethanol. We then add water, or an equal volume of water, H2O. And then, of course, with every biological test, we need to give the positive result. If fat is there, so in other words, a positive result, the solution forms an emulsion. So we get a sort of white emulsion formed. Okay, and again, the emulsion there is really important. Right, moving on. The table lists substances found in 2 types of nut, so nut A, nut B. The mass of each substance is given in milligram per gram of nut. That's important. So the units are important here. Explain the role of fiber in the squirrel's diet. So it doesn't matter whether this is a squirrel or not, just what is fiber used for. Fiber is used for peristalsis. To move food material, which is called chyme. It's called yeah, potentially chyme, but certainly like a bolus if it's in the esophagus, to move food along the gastrointestinal tract or the digestive tract. And I guess prevent sort of constipation, preventing constipation. So I've given 3 things there, but it's important to give the detail for 2 marks. Okay, explain which nut is better at helping the squirrel maintain or make red blood cells. So we need to explain why a red blood cell contains some of these things, and basically which substance is important for a red blood cell. So just very quickly, red blood cells, of course, are biconcave discs, and they basically don't contain a nucleus, no nuke. They contain lots of hemoglobin. Okay, and hemoglobin is essentially a tetramer. So there's 4 subunits, 4 protein subunits, and at the center of each, you have an iron ion, Fe2+. Okay, contained within a group called heme. So basically, this is saying, do you know hemoglobin contains iron? And if so, which of these nuts is going to be more beneficial to make red blood cells? The answer is the higher number, so nut A. So I would say nut A, because, so we need to explain it, there is more iron for making more hemoglobin to carry oxygen, which of course is the whole point. Nut A has a mass of 28 grams. Fine. Calculate the total mass of carbohydrate in the nut. Okay, so we need to go back to here now. So if nut A is 28 grams, these values here are the mass in milligrams per gram. So we need to carbohydrate. So 167 here for nut A. So we have 167 milligrams per gram of carbohydrate, of carb. Horrible handwriting. So we need this in grams in total. Okay. So first of all, I'd work out how many milligrams there are in 28 grams, because that's what we're asked to do, right? The whole nut is 28 grams. So we need to multiply it by 28. So 167 multiplied by 28 is 4,676 milligrams. And to then convert to grams, so per 28 grams, which is basically 1 nut, we need to convert this into grams. Okay, and of course, you need to know your conversions for this. So that would be 4.68 to 3 sig fig. So it'll be 4.676 grams. And we're going to give that to 3 sig fig, which would be 4.68. So whoops. Oh, where's my... Oh, there we go. 4.68 grams. Easy. Right, so graph shows the number of squirrels, foxes, and trees in a woodland between 2 time points. So just to look at this, we've got the number of organisms per kilometer squared, right? Same units, so that's fine. Same axes, and obviously 2010 to 2018. Discuss, right? That's our command words. That means we need to give sort of potentially some pros or cons, or certainly lots of different reasons why there is a decrease in the number of squirrels. Use graphs and knowledge. Right. So I'm going to use graph first. So why does the squirrels decrease? Well, we need to talk about the foxes and the trees potentially. So the foxes definitely have increased. And the trees, if I get a rough value on that, have decreased slightly. Again, use a proper ruler. I would say they've decreased a little bit, probably not a huge thing. So I will say, first of all, the number of foxes has increased. This would reduce, of course, the number of squirrels because they are predators, right? So this increases predation of the squirrels. I think that's a fair point for that. The next one that I would say is that there are, of course, fewer trees. And so I would maybe say that this obviously is a sort of habitat for squirrels, so they can climb trees to get away from foxes and that kind of thing. So squirrels can't climb to evade predation. It's also destruction potentially of their habitat. So numbers will reduce. That's what I'd say from the graph. We can now use our own knowledge as well. So why would the number of any organism, I guess, decrease? I would say that there could be migration, typically emigration of the squirrels. There could be disease, kills the squirrels. And you could, of course, say that maybe sort of humans build sort of on their habitat. The squirrels, by the way. Reducing numbers. 1, 2, 3, 4. That's 5 points. Is there anything else we could say? Not that I can think of, so I think I'm going to stick at that and not worry too much about it. If there's something on here that isn't worthy of a mark, and I haven't picked up the 5th mark, I'm not going to worry too much. That's 1 mark so far, and we're already on question 3C. That's not bad going. So let's move on. Right, question 4. Enzymes are biological molecules that act as catalysts. State what is meant by the term catalyst. Okay, this is not a biological catalyst, it's just asking for catalyst. So it is a substance, and it's worth pointing out that if this said biological catalyst, we would have to state that it's biological because it's a protein and produced in cells. Okay, but we don't have to do that because it's not asking for biological. So we just say it's a substance that speeds up the rate of reaction by lowering the activation energy without being chemically changed itself or used up, so it can be reused. Okay, so very common question that. Again, 1 mark in about 20 seconds, so there we go. State what is meant by the term metabolic. So metabolism is just chemical reactions with it. It's all of the chemical reactions within your cells. So I would say that metabolic means it's to do with metabolism. Which is the chemical reactions that occur within cells. Moving on. Okay, a little bit of highlighting required here, probably. Teacher investigates the effect of enzyme concentration. Okay, so we're going to change that. So that's the independent variable on the rate of reaction, which we're going to measure. So that's therefore the dependent variable. He uses the enzyme catalase, which is found in a potato. Okay, so that's, I mean, I don't think I care that it's in a potato, but he changes the enzyme concentration. We've already assumed that by adding different numbers of potato discs. Okay, fine. Catalase breaks down hydrogen peroxide solution into water and oxygen. So that's important. So it breaks down hydrogen peroxide into water and oxygen. Water is going to be difficult to measure here. Like, how would you measure the rate of reaction? Measuring how much water is produced is going to be difficult, but absolutely you can collect oxygen gas. So we can collect this using upward delivery. Okay, which is of course where you have a sort of water trough here, and you place your measuring cylinder like that over. You sort of have your reaction happening in a test tube. You have a bung, and the gas comes out there like that, and it will displace the water, right? So as your gas is collected, it will displace the water, and you can read off essentially the volume of gas produced. That's a horrible diagram, but hopefully gets the point across. His method is to cut the same size discs. Good, so that's a control variable from a potato. What it's not saying is this potato should therefore be the same species and age of potato. That needs to be the same. 5 centimeters cubed of hydrogen peroxide solution. It's not giving us a concentration, so I'm going to highlight that and say conc. Into 5 test tubes. Add a different number of potato discs, but they're the same size, so that's fine. And we're going to measure the volume of oxygen gas produced in the same time, so that's fine as well. So what I'm going to query here is, are we using the same species or age type of potato? And the concentration of the hydrogen peroxide should be the same. No, sorry, correct. Sorry, correction. I've just read that again. It's the effect of enzyme concentration. So actually that is going to change, isn't it? We're changing, that's what we're changing, so that's fine. So we repeat it 4 times, good. Calculates a mean rate of oxygen production. So the results are shown here. So we've got enzyme concentration and the mean rate of oxygen. Right, plot a line graph, to show the effect of the enzyme concentration on the mean rate. So don't forget dependent variable here, independent variable here. So dependent is going to be the mean rate of oxygen production. And that is going to be centimeters cubed per minute as units. And then we've got enzyme concentration, number of potato discs. So we haven't got an actual concentration. But we've got number of discs. So number of discs is 2, 4, 6, 8, 10, nice and easy. So we can just go in ones here, I think. Assuming we get 10, 1, 2, 3, 4, 5, 6, yeah. So 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. Uses up a nice proportion of the graph. And dependent variable, we go from 2 to 8.2. So we'll have to go up in 1, 2, 3, 4, 5, 6. Yeah, probably the same, nice and easy. So 1, 2, 3, 4. Oops, missed one out. Easily done. 3, 4, 5, 6, 7, 8, 9, 10. There we go. Right, so we just need to plot this now, which is a little boring, but there we go. So 2 is 2. That's nice and easy. So 2 and 2. 4 is 4.4. So 4.24. There we go. Oops, I've done that completely the wrong thing. Sorry. Oh no, that was correct. Sorry. Oh, this thing driving me nuts. Right, here we go. So 2 and 2. So 4 and 4.4. So 4.24. There we go. So that's the first two done. 6 is 7. So nice and easy. So 6 is 7. Again, you'd be more accurate on paper. It's quite difficult on a surface. Ugh. There we go. 8, 8.2. So 8, 8.2. And then 10 is 8.2 as well. So that's the same. And it says draw with a straight line. Do you go through the origin at 0? The answer is, well, yes, because if you have no enzyme, then we can assume that the rate would be 0. That it's an enzyme catalyzed reaction, so it wouldn't happen. So you can absolutely go through the excuse me, go through the origin. Sorry, dying. Right, so it's not going to be the best, but we can do this. There we go. And again, just to make you aware of how these graphs are marked, best thing to do is this, which is called slap K. So this is for scale. Okay, is it appropriate? Well, yes, it is, and it covers at least half the graph. The lines, so make sure your lines are drawn appropriately. So either a curve of best fit or a line of best fit or a straight line in this case between points. Your axes, so axes labeling. So this is the labeling, that's correct. So independent, dependent on the right thing with mean rate of oxygen production and enzyme concentration, and axis units. So they are on there as well. Finally, the points plotted correctly and accurately, yes. And then the only other thing is K, which stands for a key. And we don't need a key because we've only got one line. But if you had multiple lines, you'd have to draw a key somewhere and differentiate between the two. Okay, but that's where your 5 marks come from for a graph. Explain the effect of increasing enzyme concentration on the rate of oxygen production. Nice question, this, because it allows me to, first of all, it's an explain. We have to talk about increasing enzyme concentration on the rate of oxygen production, so the rate of product. But it says explain the effect. So we have to tell them what the effect is. And of course, you can see on your graph that at 8 potato discs, we have leveling off of our graph. So we're going to have to say that as well. Okay, so I would say initially, as enzyme concentration, I'll abbreviate here, increases, so does, or the rate of oxygen production also increases. Okay, until 8 potato discs, where the rate of oxygen production plateaus or levels off at, and I'm going to give a value here, whatever it was, 8.2 centimeters cubed per minute. Okay, so again, give data That's really important. Give data for these questions. We haven't explained why this is true, though. So this is true because you have more enzyme active sites available, therefore an increased number of collisions between your active site and substrate. Okay. Forming, again, give detail, enzyme substrate complexes. There we go. However, we haven't explained why it levels off. It levels off because if you imagine here, right, imagine you've got your enzymes like this, and you've got your substrate, right? You've got 1, 2, 3, 4, 5, 6, like that. All of these can be acted upon by the enzyme, right? So all of these active sites can be bound by substrate, okay? And you have some substrate left over. So therefore, these enzymes are working at maximum rate. If you were to increase enzyme concentration up here by a lot, right, you essentially run out of substrate, right? So imagine you've converted all these, you've got this substrate left, you've now ran out of substrate. So essentially adding more enzymes, adding more active sites isn't going to help you because you're already binding the maximum amount of substrate. Okay, so we need to explain that by saying the rate levels off as the substrate concentration becomes the limiting factor. And all substrate is therefore being converted into product. Okay. Now, quite a lot for 3 marks, but I don't know where the 3 marks are coming from exactly. So, you know, it's worth pointing that out. Name a piece of apparatus suitable for measuring the volume of oxygen produced. So this is a upturned measuring cylinder, so upward delivery tube. And measuring cylinder. Inverted in water. You could, if you're a chemist, of course, use a gas syringe. That's also an option. Explain why it is important for the teacher to keep the volume and concentration of hydrogen peroxide constant. So we did need to keep that constant. Yes, because we're using potato discs, aren't we? So we do need to keep that constant. So I was right. So this is to obviously act as control variables. Act as control variables. Because, and again, we need to explain why both of these, so both the volume and the concentration of hydrogen peroxide, bear in mind this is a substrate for the reaction, will affect the rate of the reaction. Name another variable that teachers should keep constant. Well, if we're talking about enzymes, right, you have to keep the temperature the same or the pH the same, right? Both of these things affect enzyme activity. So I'm going to say pH, but you could probably have temperature as well there. Right, so we're on question 5. So we're not doing too badly. I'm just going to check time. So how long have we been going? 34 minutes, not too badly. So 34 minutes, we're already on question 5. So we're doing quite well here. Right. So farmers can increase their crop yield by growing crops in a temperature-controlled glasshouse. explain how the temperature increase can result in increased yield. So you've got to think about what these crop plants will be doing, and they'll be doing photosynthesis, of course. And for photosynthesis, you need enzymes. Okay, so I would say that it allows the optimum temperature for enzymes involved in photosynthesis. So the rate of photosynthesis increases. So more, and again, we have to lead this to growth, right? So more glucose is made in the leaves, which can be used for respiration, and therefore ATP is produced for growth. There we go. Nice answer for 2 marks. Farmers sometimes use a type of heater that burns gas or oil. Okay, so we use a heater to raise the temperature. Explain why you use this type of heater to improve the yield. So this links back to the sort of oil and gas heaters we talked about, or certainly I do with removes, which is that these produce lots of carbon dioxide as a result. So I'm going to say that these heaters release lots of carbon dioxide as a kind of byproduct. And of course, why is that important for the yield? It's because carbon dioxide is needed as a substrate for photosynthesis. Right, some farmers add chemical fertilizers to their crops. Different minerals are added to chemical fertilizers. So remember, these are things like nitrates, potassium salts, phosphates. Explain one mineral that should be added to these fertilizers. So I'm going to say nitrates. So NO3 minus. And why? So I need to explain it's used as a nitrogen source for making amino acids and therefore proteins, which are needed for growth and repair. So there we go. Right, so this is a discuss for 5 marks. So discuss, so we need pros and cons potentially. Why some farmers limit chemical fertilizers. So of course we've talked about this. They'll use natural ones, so things like manure, so cow manure, etc. We need to, okay, so we're not giving both sides the argument here. We need to discuss why they limit it, essentially why they're bad. And of course, the main thing that I'm thinking immediately is leaching and eutrophication. So I'm going to say it stops leaching of fertilizer into water sources, things like lakes and ponds. This will stop eutrophication okay. So just to be clear, this will decrease algal blooms, which will mean plants can photosynthesize, and essentially fish and aquatic life can survive essentially. So plants can photosynthesize. That's because they don't die, not die from lack of sunlight. You could also say things like cow manure is cheaper, and perhaps, well, yeah, we'll say cheaper. So therefore, farmers can make more money. So how many? Is there anything else I can think of? I guess chemical things are expensive. Potentially ethical reasons with sort of so it prevents bioaccumulation and magnification. So it doesn't harm other organisms. I think we'll give that there. Yeah. I can't think of anything else off the top of my head immediately, but I think that's probably enough. We said at least 4 things there. I think chemical fertilizers. Yeah. Moving on. Right, question 6. Doing okay for time. The human liver produces bile. Explain the role of bile in digestion. Right, so for 4 marks, gosh, just give as much detail as you can here. So bile is alkaline, so neutralizes stomach acid. Again, to explain it, so the rest of the digestive tract, so things like the ileum, are not harmed. They, of course, emulsify fats of large fat globules into smaller ones. And of course, this increases the surface area for the action of pancreatic lipase. Okay. Anything else that bile salts do? Neutralize fats? No, it's pretty much it. Right, there are 3 main blood vessels attached to the liver. So hepatic artery, hepatic vein, hepatic portal vein. It's worth drawing this out. So if you have your liver here, and you've got your heart here, so the blood going to the liver, of course, will be the hepatic artery. The blood coming back to the heart is a hepatic vein, and the hepatic portal vein comes immediately from the stomach and the digestive tract. Okay, so that's there, hepatic portal vein. So I would draw that out in the exam, just to be clear. So how many of these blood vessels transport deoxygenated blood from the liver? The answer is 1. Okay, it's the hepatic artery. Nice and easy. Right, a little bit of wordy stuff going on here. A scientist investigates the glucose concentration in a person's hepatic portal vein. Okay, so again, going back to my diagram here, we're talking about glucose here. After the person has eaten a meal, that's interesting. Okay, so immediately I'm now thinking, oh, this could be about sort of insulin and blood glucose, right? So insulin is released by the pancreas, which decreases the blood glucose levels, don't forget, after you've eaten a Snickers bar or something. So the scientist method, they give a person a meal to eat. Okay, we don't really have much detail on the meal. After 1 hour, okay, is that really enough time? Take a blood sample from the hepatic portal vein and a blood sample from the hepatic vein. Okay, so we're talking about these two, aren't we? So hepatic vein and hepatic portal vein. Okay, so after you eat a meal, you'd expect lots of food particles lots of food particles here to be absorbed by the ileum, etc., transported here through the hepatic portal vein to the liver, where it then gets obviously detoxified in the liver. Measure the glucose concentration in both samples. After 5 hours, take another blood sample from each of the two veins. Right, okay. So 1 hour and 5 hours basically from each of these. After 1 hour, the concentration of glucose in the hepatic portal vein is higher than the concentration of glucose in the hepatic vein. Okay, yeah, that makes sense. Explain the difference in concentration. So why does the hepatic portal vein have a higher concentration of glucose after the meal? It's because you've got lots of glucose in your stomach that's being absorbed by your ileum and being transported to the liver. And obviously within 1 hour, it's not long enough for the liver to then deal with all that stuff and get all of it. So a lot of it will be, whoops, what has happened here? My OneNote appears to be crashing. Oh dear. Okay, it's just reloading. Can you see that? Hello? No, I need to present again, do I? Share screen. Oh, it's annoying. Right, there we go. Hopefully we're back. Just let me check. We are back. Yes, we are. Okay, so explain the difference. That is because, of course, you have eaten or you've eaten a meal where the glucose has been absorbed in the ileum into the blood, specifically the hepatic portal vein. Now, of course, what happens in the liver, why else would it be higher than the hepatic vein? Of course, any glucose that comes into here doesn't immediately get kicked out because the liver will metabolize it, won't it? So the question is asking, what does the liver do to the glucose? So, of course, that's the site where insulin acts primarily. So insulin is released by the pancreas, and in the liver causes glucose to be converted to the storage polysaccharide of glycogen, which would be another reason why the hepatic vein has less glucose in it. After 5 hours, the concentration of glucose in the hepatic vein is higher than the concentration. Okay, well, you've stopped eating, right? You've stopped absorbing. So it's the opposite, less insulin, basically. So you have less glucose in the ileum being absorbed into the hepatic portal vein. Therefore, you have less insulin released. We have less glycogen. Actually, well, it wouldn't be less. Would it be no insulin if you're not eating it 5 hours afterwards? To be honest, no insulin. Therefore, why would it be higher? So glucose is being converted from glycogen, isn't it? So glycogen is being broken down into glucose in the liver. Therefore, the hepatic vein has an increased concentration of glucose. Handwriting's getting a bit poor. Right, so we're on question 7. I forget how many questions there actually are in this. The 10, maybe? Yeah. So we're actually not doing too badly. So 7 out of 10. Question 7 out of 10. And how long have we been going for? Time check. 49 minutes. So we're actually doing pretty well. We're less than an hour and we're already 2/3 of the way through easily. Right. So fruit flies. So we've got a male fruit fly with long wings and a female with long wings. Okay. So they've both got long wings. Wing length is controlled by a single gene with 2 alleles. The dominant allele, which is big L, produces long wings. The recessive produces short wings. When we mate the 2 fruit flies, this is what we get. Interestingly, we get the recombinant types. Okay. We get males with short wings and females with short wings. So they, if we're talking about alleles, they must be little l, little l. And in order to get little l, one of each parent. So if you imagine your sort of Punnett square, to get little l as an option, both parents have to have a little l in their gametes. But of course, you get the others as well, so they therefore must be big L here, like that. So they're both heterozygous. So genotypes, both heterozygous A. Genetic diagram, it shows sex linkage. No, sorry, how sex is inherited. Yeah. So complete genetic diagram to show that equal numbers of male and female offspring are produced. We're not told about X and Y's, so we can assume that it's the same as humans. So X and Y is male, X and X is female. Complete the genetic diagram to show that equal numbers of male and female are produced. Well, those are the gametes for the male, so the female must give X and X. So it's X, 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 Y, X, 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 Y. And we want to show that equal numbers are here. So what I might then do in an exam is just to highlight that and go, they are male, and then maybe highlight those and go female, just to be super clear. Explain why the results of the cross, so explain, might not produce exactly equal quantities. So this is about, okay, well, what happens during fertilization? And of course, it's random, right? So these gametes, exactly which of these 4 outcomes here that you get is completely random each time a mating happens. Okay, so we have random fertilization. Okay, random fertilization of gametes. During, okay, I don't know where the second mark's going to come from here. And do they do crossing over? I'm assuming they probably. Well, actually, crossing over won't matter. This is the first one. This is the first question where I'm thinking, I don't really know. I don't know. I'm going to star that question. That's the first one of interest to me. And I'm going to do what I advise you to do in the exam, which is about now. I've spent too much time thinking about this question, so I'm going to call it a write-off mark. And if I only get 1 out of 2, so be it. It's 1 mark at the end of the day. And I'm on question 7, so I'm not going to waste my time. Okay, so this is a Corms question. There's always a Corms question in these papers. So fruit flies, so that's the organism we're going to be using, feed on decomposing fruit. The smell of the fruit attracts the flies. Design an investigation to find out if the smell of decomposing apples attracts fruit than decomposing bananas. So we're going to have apples and bananas. We want to make sure that they're decomposing to the same extent. So what am I going to change, first of all? And this is how I would set out a Corms question, by the way. I would go change, so C. I would set up 2 boxes that are interconnected. And in each box, place the same biomass of decomposing apples and bananas. The organisms I would use fruit flies, so use the same species, age, and potentially sex of fruit flies. And I would maybe use, I don't know, you might want to use 100, something like that. R, I would repeat, although I'm going to put that at the end actually. So M, so what am I going to measure? So measure 1, I'm going to record the number of fruit flies on both the apple and banana. Actually, in the boxes. Okay, so I'm going to cut off the 2 boxes after a set time point. So measurement 2, I'm going to do this after a set time. So maybe after 1 hour, something like that. So what am I going to keep the same? I'm going to control the age, the sex, the number, the species of fly. I'm going to control the biomass of apples and bananas, which I've already said, but can say it again. I'm going to control maybe the temperature of the boxes, make sure that's the same. And of course, the number of flies. So I'm going to use maybe 100 flies. Finally, I'm going to repeat the experiment at least 3 times. Calculate an average maybe. There we go. Okay, so nice and easy, 6 marks in about 2 minutes. This is absolutely how I would set up your Corms question, by the way. So long as you write in good English, you can absolutely just write it like this. Cool. Moving on. Don't waste time with the Corms, right? Just literally set it out like that. For repeat, don't forget it. Just put it at the end. Easiest thing to do. Instead of saying, "Oh, I would take 100 flies and I'll do that 3 times." Just at the very end of your experiment, just say, "I would repeat the experiment 3 times," or however many times you want to do. Right. Question 8. We're nearly getting there. We're probably about an hour through now, so half time, and we're already nearly getting there. So, right. Question 8. Diagram shows a section through a leaf with different parts labeled P to U. So the magnification is 100 times. Which part of the leaf transports amino acids? So we're going to be looking for the phloem here, which is in the vascular bundle. So that's in S, right? It's not differentiating between the 2 here. So, of course, the phloem would be sort of at the bottom if it was differentiating, but it's not. So the answer is it's just S, right? Moving on. Which part absorbs the most sunlight? So which part absorbs the most sunlight? So absorbing the most sunlight, most of it will pass through the waxy cuticle and the upper epidermis. So it'll be the palisade mesophyll cells, which are at the top because they have lots of chlorophyll, of course. So the answer is R, or at least that's what I'm going to go for. Which part transports the products of photosynthesis? That's S again, isn't it? Because the phloem transports glucose or sucrose actually as well. So that's S again. So the answer there is C, S. Which part reduces the amount of water escaping? So this is, of course, the stomata. So what options have we got? P, Q, R, S. Ooh. So, okay, so it's not pointing to the stomata. There's nothing labeling the stomata. Oh, sorry, reduces water loss. Escaping, that's P, isn't it? Because it's Ooh, actually, is it? Which part? Why am I being confused by this? Which part? Yes, P. Yeah. It's the waxy cuticle here. Yeah. So P, which is A. There we go. Moving on. Explain. So another explain question. How the structure and positions. We have to talk about. So important. Talk about both of these of R and T cells, which are the two types of mesophyll, right? You've got your palisade mesophyll, and you've got in T your spongy mesophyll. Oh, sorry, my OneNote's crashing again. This is so annoying. Are we back? No. Sorry, I hope this is not going to happen again, but who knows? Right, share that again. Right, we're back. So explain how basically palisade mesophyll cells and spongy mesophyll cells are adapted for photosynthesis in terms of structure and position. So I would go R are the palisade mesophyll cells. And they are towards the top of the leaf for maximum sunlight absorption. They contain lots of chlorophyll for absorbing sunlight. So absorbing sunlight, same thing. And that they are densely packed together. Again, to increase the surface area and sunlight absorption, which I won't write again. T, these are the spongy. Sorry, my handwriting is getting worse. Spongy mesophyll cells. And again, these are towards the bottom of the leaf. Why is that useful? Of course, it's to absorb carbon dioxide through the stomata. So spongy mesophyll cells, bottom of the leaf. This means that the carbon dioxide absorbed via the stomata, which are also at the bottom of the leaf, can be taken up and transported to the leaf for photosynthesis, of course. They are less densely packed. And this is, of course, for diffusion of oxygen and carbon dioxide for gas exchange. There we go. So 4 marks, again, in probably 2 minutes. So, you know, this is where you save time, people, right? It's knowing your biology, right? You know, question 8 there, we've got hopefully 1, 2, 3, 4, 5, 6, we've got 8 marks there in probably about 3 minutes. So I've probably saved myself 4 or 5 minutes at least. Right. Okay, this will be tricky for me, I think, unless I can do it with arbitrary units, because you need a ruler. So it's magnification. We want the actual thickness of the leaf. So remember, magnification is image over actual in terms of your triangle. If you want to draw your triangle of power, magnification is image over actual. So we want the actual thickness of the leaf. So it's image, so it's AIM. So actual is image over magnification, right? That's the equation we want. So we're going to be dividing something by 100, and it's the actual image. It's in millimeters, people, right? Be clear about your units, right? Read the question. It's in millimeters. So I need my image size. I need to measure this in millimeters. And is it the width that I want? Thickness of the leaf. So this way. So thickness of the leaf, it tells me which way. So I need to measure, right, how do I do this? Draw, ruler. Like I said, this the only thing is it doesn't have, that's annoying, it doesn't have actual units, but hopefully that's about right. So I might be slightly off here, by the way, but it's 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 18, 19 and a half. Be very specific. Okay. Now, I don't know what the units are of that ruler, which is annoying. It's sort of arbitrary units. So I'm hoping this might still, actually, it's not going to work, is it? Okay, you'd have to do that in your own, whatever that is, by the way, in millimeters, okay, is what you'd use. I'm going to use 19.5, just to make it easy for you. So I would do 19.5, assuming that that's in millimeters, divided by 100. Actually, that probably looks like it's in centimeters, doesn't it? Yeah. Okay, well, let's give it a go. 19.5 divided by 100 is what? 0.195. In millimeters. I'm assuming that's in millimeters. So that's probably going to be wrong, because this value is incorrect, because I can't measure that on a Surface device. So you'd have to use a ruler for that. But whatever value that is, that's what you plug in. So there we go. Right. We've got some water lilies here. You need to know water lilies because you need to know that these are an example of hydrophytes. Oops. So they are adapted for living in high water conditions, as opposed to xerophytes, things like cactus, cacti and marram grass and things, which survive in low water conditions. These are the opposite. And they have stomata. One of their adaptations is that they have stomata on their upper surface as opposed to their lower. And that's because their lower surface, of course, is submerged in water. So suggest why the leaves only have stomata on the upper surface. It's because the lower surface is in the water, and the upper surface is exposed to the air, which is where carbon dioxide can therefore be absorbed from. So that's what I would say there. Of course, it also allows transpiration as well, as evaporation of water molecules. So it allows transpiration. Again, I'm giving detail. I'm not quite sure where the mark scheme is going to go with this. So allows transpiration as water molecules can evaporate essentially into the atmosphere. Whereas, of course, they can't do that if they're submerged in water. So only 2 marks, but I don't know where the 2 marks are coming from. So I'm just giving detail there. And I've got the time to do it as well. Let's have a drink. Right, explain the role of lymphocytes in the immune system. So lymphocytes, you don't need to know a huge amount of detail at GCSE. You may know that these are called B and T cells. You don't need to know anything really about the T cells, but you do need to know that B cells produce antibodies that look like this. And you need to know that antibodies do 3 things. They opsonize, so they act as opsonins, so they essentially tag pathogens. They cause agglutination, and they can act as antitoxins. Okay, and they do all this because they bind to antigens on the surface of a bacterium or a pathogen. So if you have bacteria like this, the bacterium has sort of like antigens on its surface. So these are molecules that cause immune responses. So these are antigens. Any molecule, that's the definition, any molecule that causes an immune response. And your antibodies have a complementary shape to the antigens, and they can bind meaning that the antigens can't then evoke an immune response. And therefore or of course, if the bacterium produces toxins, you can have other antibodies that maybe can bind to the toxins, stopping the toxins from doing their job. And the antibodies can stick together. So you've got lots of bacteria here sticking together because you've got lots of antibodies bound to them, bound to the antigens here, and the antibodies themselves can stick together, causing like a agglutination, which means that these can be dealt with by phagocytosis. So those are your three things. So what's the role of them? Well, the B cells produce antibodies. Of course, what I forgot to say is that they can produce B memory cells as well. So memory, which I'll talk about in a minute. Important in vaccines, of course. So B cells produce antibodies. Probably worth saying what the antibodies do. So antibodies are specific for a given antigen on a pathogen. I'll put that on a pathogen. And they can cause agglutination of pathogens. They can tag pathogens or opsonize them, and they can act as antitoxins. Oops. They can also act as B memory cells, which is important in vaccinations to have a secondary immune response. Okay. Cool. That's probably enough. Right, phagocytes ingest bacteria. Explain what happens to bacteria after they've been ingested by phagocytes. Well, they're broken down by hydrolytic or digestive enzymes, and then the breakdown products are absorbed by phagocytes or by the phagocyte itself. I can't really think of much else to say there. So yeah, I think that's probably fine. Right, I've got lots of stuff to read here. They always put this towards the end, by the way, when you're getting tired. So it's worth sort of thinking about that. I mean, what question are we on? 9. We've literally got a few pages to go. So if you have got time at this point, it might be worth just sort of taking a minute or two just to have a drink and have a breather. What time are we on, actually? We've been going for an hour and 13 minutes, so we've got 45 minutes or so left to do not very much. So really not very much. What 1, 2, 3, 4, 5, yeah, like 5 questions. So pretty good. Right, a scientist uses this method to investigate how vitamin C affects ingestion of bacteria by phagocytes. Different concentrations of vitamin C add distilled water to a 1/4, so that's absolutely a control. So that's basically no vitamin C. So it's a control experiment. Not a control variable, it's a control experiment, which I now talked about in class. We add phagocytes. Okay, I would say how many number? Leave the tubes for 1 day. Okay, fair enough. Add 10 centimeters cubed of culture of non-pathogenic bacteria, good, so it's not going to harm us, to each tube. Keep at 35 degrees C. That's not physiological temperature, so 37 degrees C, why not? 37 degrees C is kind of the human internal body temperature. Measure the number of bacteria ingested. Okay, difficult, potentially. So these are the results. So we've got concentration of vitamin C in arbitrary units. So essentially going from 0.5 here to 1, we've doubled it. Number of bacteria ingested in 2 hours. I thought they left it for. Oh yeah, they left it for a day. In 2 hours, right? Okay. Is that long enough? Maybe. Which of these is the rate of ingestion of bacteria in the control? Oh, I see. Right. Okay. Yeah. So basically, this is testing. Is the what is the control? It's no vitamin C, so it's 0. There it's what 100 and well actually hold on. What would be halfway between 150 plus 200 divided by 2, so that's 250. Actually no, hold on, no it isn't. 25, isn't it? So 175. Okay, so the rate, so in 2 hours, it would be 150 to 175. 175 minus 150, 25, divided by 1, 2, 3, 4, 5. So each one of these is worth 5. So 155, 60, 65, 70, 75. So that's 155, 150, 160, 165, 170. So this value here is 170 in 2 hours. So per hour, it's 170 divided by 2, isn't it? So that's 85. Oops. There we go. Nice and easy. So that's the rate, just to be clear. State the independent variable in the investigation. Well, that's the concentration of vitamin C, because that's what we're changing. Independent variable on the x-axis, of course. State 2 biotic variables. Okay, so biotic is living. So what living things do we need to control? So we need to look back up here. I think the obvious one is the number of bacteria and the species of bacteria that have been added here. So number, species. Also the phagocytes, again, number or concentration, perhaps. So I think they're the most obvious things. So the concentration of phagocytes and the species potentially of bacteria added. Scientist concludes that vitamin C will protect people from pathogens. Comment on the conclusion. So why is it good and why is it bad? Sorry, why is it correct? Why is it not? So vitamin C does protect people from pathogens. It does increase killing, as you can see from the graph. So I'm going to say, well, vitamin C does increase engulfment of pathogens by phagocytes. Okay, it's a general positive relationship. So that's good. But I mean, I can't think of anything else good. So bad, well, it's not done at 37 degrees C, because this is talking about people. Yeah. So people that are at 37 degrees. So this is not done at 37 degrees C, which is the internal body temperature of a human. Were there any repeats? So actually, it would help you if you think about corms, actually, here. If you were designing the experiment, have they ticked off everything with corms? Well, no, they haven't, because they haven't repeated it. So no repeats. Anything else here? Vitamin C, person's diet will protect people. Okay. Well, it's not even done in a human, is it? Right. it's all well and good saying that. This is done in a test tube, and actually also pathogens. Do you know what a pathogen is? Right, pathogens can be bacteria, but they can also be fungi, right, and they can be viruses, right, and various other things. So actually, two other things here. It's not done what we call in vivo, right? It's been done in a lab. And I would say that bacteria are not the only pathogens. Oh, sorry, OneNote crashing again. So annoying. Right, we're back. Sorry. I don't know why it keeps crashing today. It's not, it's sort of not doing very well. Right, so that's basically what I would do there. So comment on means give pros and cons. Although I've just completely lost what I've written. So no repeats. It's not done in vivo. What was the other thing? Bacteria are not the only pathogens. Right, I think that's probably enough for 4 marks there. Again, right, question 10, final question. Here we go. Diagram shows some of the structures involved in fertilization of a flowering plant. Okay, some of you will realize that there's actually 2 nuclei here. That is correct because they actually undergo something called double fertilization, which I did talk about, but you don't need to worry too much about it. So don't be put off. It's just nuclei. Name A, B, and C. Well, of course, this whole thing is the carpel. So the A is the stigma, B is the style, and C, the structure, is the ovary that obviously contains the oocytes. On the diagram, draw the path the pollen tube takes after the pollen grain has germinated. So we'll have the pollen tube going down here and into the micropyle there. And if you're interested with why, it's because you have certain cells, let's say this one in blue, let's say this one in blue here, that actually releases certain sort of chemical attractants, if you like, that attract the pollen tube. So that's, yeah, a little bit of extra for you if you're interested. Right, describe what happens to part C after fertilization. Okay, so C is the ovary. So we need to talk about itself, and of course the ovary. So this is just factual recall here, only for 2 marks, which is that the ovule becomes a seed, and of course the ovary becomes the fruit. That's 2 things for 2 marks. Right. A scientist investigates the effect of 4 different storage conditions, germination. Right, 500 seeds for 2 months. She counts the number of seeds that germinate when planted. Calculate the percentage increase. Right, so percentage increase, people. I'm thinking cuckoo, change over original. That germinate in wet and cold conditions, so that's that one, compared to the number of seeds in wet and warm, so that one. Okay, so we can forget these 2. So 476 to 432. Okay, so that's what I'm interested in. So just to highlight those 2 values, there we go. So change over original, right? So the change is 476 minus 432. Okay, do the change from the start to the finish, right? And whatever value you come up with will give you the right sign. So don't flip that just because you're going to end up with a, if you were going to end up with a negative number, don't flip it. Over the original, which is of course 476, and its percentage, so I need to multiply by 100, and that will hopefully give me the answer of, if I plonk this into my calculator, 476 We'll take away 432. Oops. 476. Take away 432 over 476 times 100 gives me 9. Oh, hold on, is that right? Is that right? That doesn't seem right in my head. Have I done something wrong? Hold on, right, let's start again. Change over original. I think I got a weird answer, which doesn't make sense. Minus number, which it shouldn't be. Calculate the percentage increase in the number of seeds that germinate in wet and cold conditions. Oh, done it the other way around. Right, there you go. I knew something had gone wrong. Go that way. Oops. Percentage increase. Yeah. So again, percentage increase, right? So so we're increasing. So we have to go that way. So again, I would do 476. Whoops. Take away 432 over 432. Times 100. That might give me a better looking answer. 476 minus 432 over 432, which should, and then times 100 give me a positive. Oh yeah, it does. Okay, good. So that's 10 what is happening to my surface? I think my surface is dying, but right, 10.2%. Okay, always worth checking your answers. If it says percentage increase and you end up with a decrease, double check your answers. Which so I nearly fell into that trap, right, just by not reading the question properly. I thought that was a bit easy for question 10, right? If something like this is in question 10, it's probably going to be a bit more complicated. So, right, final question, or 2 questions. Suggest why fewer seeds. So suggest, I don't need to have been taught this, why fewer seeds germinate when they're stored in wet and warm conditions than in the other conditions. When they're stored in wet and warm conditions. Well, they're already germinated, weren't they? Assuming there's water. Yeah. So they're wet and warm. Assuming there's oxygen and things around, they'll have already germinated. As the wet conditions will have activated enzymes needed for germination. Give one observation that shows that a seed has germinated. Well, of course, you end up with a radical and a plumule. Are seen, right? So again, from your seed, you'll end up with a sort of plumule starting to form leaves, and you'll end up with a radical here forming the roots. Right, that's it. So in terms of time, battery's running low as well. Right, I'm going to pause the video and get the mark scheme up in a minute. In terms of time, that's 1 hour 28 minutes. Okay, so I'm just going to go up to the top here. It's a long paper. Long paper. So before I get the mark scheme and mark it, 1 hour 28 minutes. Not bad to say that I explained a little bit as well. So that gives me a solid half an hour to now go through and check that I have answered everything correctly, that I double check my calculations, double check my units, double check definitions to make sure you've got enough detail in there. So I've got half an hour to do that. I'm not going to do that. I'm just going to mark it and see where we go. Potentially, I might have lost 1 or 2 marks. I think there's certainly a couple of questions where I wasn't quite sure. There's the write-off mark, which will be interesting to see. But hopefully you get the point that, you know, I won't necessarily get every mark on a paper, and, you know, use it as a learning opportunity. But hopefully, you know, I've got enough marks to easily secure the 9. So, right, I'm just going to get the mark scheme up and I'll be back in a minute. Right, I'm just going to check that we are recording, which we are. Good. Okay, so I've got the mark scheme up. We're going to go through it and see how I did. So A is B, good. Next. Secondary consumers is C. Next. Eyesight, seeing, visual optic, yes, 1 mark. 17.7, yep, so 5, yep, good. So 530, yeah. 100, sorry, 17. I now can't move this down weirdly. Right, anaerobic respiration provides energy ATP without oxygen. So anaerobic respiration, ATP, absence of oxygen. Easy, 3 out of 2. Good, so we're doing well so far. Right, has a nucleus. Yeah, correct. Mitochondria or organelles, yes. Circular chromosomes. Have chromosomes more than 1 chromosome? Oh, prokaryotes have a circular chromosome loop of DNA. Yeah, good. So 3 marks there. Plasmodium and malaria, good. 2 marks there. It does. Yeah, so trypanosoma and sleeping sickness. Yeah, good. So crush or grind the nuts. Yeah. Add ethanol. Yeah. Add water. Yes. And emulsion formed. Yes. So easy, 3 marks there. So 4 marks out of 3. Nice. Explain the role of fiber. So peristalsis, moving food along the intestine. Yeah, digestive tract. Right, 2 marks there. Nice. Prevents constipation is also on the mark scheme, so that's good. Nut A, because more iron for making hemoglobin. Yeah, more iron, hemoglobin. Bingo. Nut A, so the mass is 4.68. Good. Whoops, didn't mean to do that. So 2 marks there. Good. We're full steam ahead. So this is for foxes. So more foxes to eat the squirrels or increase predation. So annoying, only 1 mark for all of that. That's annoying. Foxes eat nuts, so less food. Oh, of course they do. Didn't put that. Drat. It even showed a picture of the squirrel eating a nut. Okay, never mind. Learning opportunity there. Good. Trees, yes. Disease or infection, yeah. Migration, yeah. Humans build on squirrel habitat. Oh, human impact, yeah, good. Right, easy, good. 1, 2, 3, 4, good. 5 marks. Worth bearing in mind the other points here. Time of year when the numbers are counted, or the weather conditions. Interesting. Or the fox increases more than the tree decrease. Oh well, there's enough to say there for 5. A very common question, this, by the way, is to discuss reasons why population numbers change. It's all about, you know, disease, migration, human impact, deforestation, that kind of thing, as well as predation. Right, number 4, substance that speeds up chemical reactions. Oh, okay, that was easy. I gave quite a long answer to that, but never mind. Metabolic reactions are chemical reactions that occur within cells. Yeah. Right, the graph. Oops. So scale, linear, half of grid. Yes. So that's scale, sorry, there. Yeah. Draw neatly between points. Yes. Axes are the right way round. Yes. Points plotted correctly. Yes. Axes are labeled. Number of discs and oxygen production with the units. Yes. Good. 5. Explain the effect of enzyme concentration increasing. Oxygen production increases. Yes. Up to 8 discs, which is 8 or 8.2 centimeters. Yeah. So again, quote the units, give the data, quote the units. More enzyme available to react with or break down hydrogen peroxide or enzyme substrate complexes formed. Yes. Well, that's 3. There we go. Until all substrate molecules are consumed, substrate becomes limiting. So we said that as well. But yeah, give the detail for those questions. Name a piece of apparatus, gas syringe or measuring cylinder. Yeah, good. Act as a control variable Only one variable has changed, one independent variable. Control variable, there we go. These also affect the change of rate. Good. And then pH. Good. We're on 100% so far on question 5. Pretty good going. Temperature increases, so does the rate of photosynthesis. Yes. Increased kinetic energy or energy of molecules, more collisions. Didn't say that. Oh, so this is wanting collision theory. Ooh. I guess it would with temperature, perhaps. So, okay, good learning opportunity there. It wants collision theory. Have I lost a mark then? Optimum. Oh, I didn't say optimum. Oh, yeah, I did optimum temperature. Yeah. Okay, so I didn't. Good. Worth bearing in mind, though. Release lots of carbon dioxide. Yes, because it's needed as a substrate for photosynthesis. Yes, there is a reactant. Good. We could have had nitrates for amino acids. That's the obvious one. Of course, magnesium for chloroplasts as well. Phosphates would be for DNA and RNA. So that's fine. Right. Was this a right? No, this wasn't the write-off. Increased soil concentration. Oh, of course it does. Increasing the concentration of chemicals and nitrates and phosphates in the soil will reduce the water potential of the soil. So therefore, less water will be taken by osmosis into the roots. So roots and osmosis. Okay, learning opportunity there. Let's see how many we got though. Leaching, yes. Eutrophication, yes. Algal bloom, yes, that's a mark. Oh, algal bloom is a mark. There you go. Give detail, people. I've scored there for giving detail. Loss of aquatic life, water plants die. I guess I've said the opposite. I've said that they survive because we don't get all this. So okay, that's fine. That's a mark there. Cow manure is cheaper. Can produce organic, natural. Natural fertilizer, absolutely. So I've got 5. Nice. I don't know about the can produce organic, earn more money. Oh, I see. Because you can make. Oh, okay. Yeah, I think that's that point. Bioaccumulation magnification. Interesting. That's not on there. Huh. So, interesting. I guess because it impacts the farmers about. I don't know. I think that should be a valid point, but oh well, we've got 5, so move on. Right, liver. Neutralize stomach acid. Good. Key word there, neutralizing. You need to have it. Optimum pH for enzymes. Oh, didn't put that. That's a good point. To allow the optimum temperature for enzymes. So your lipases and proteases and things. Yes, that's a good point. Emulsification of fats into smaller droplets. Yes, for increasing surface area. Good. Okay, so we easily got 4 there, but that's a really important point. Definitely make a note of that. Right. So we're still on 100%, which is good. B is the correct answer. Good. Explain the difference in concentration. Glucose absorbed, taken in, removed from the blood. Yes. Insulin and glycogen. If you've mentioned insulin and the fact that we get more glycogen, right, 3 marks, done. Nice and easy. This is the opposite. So no insulin, glycogen to glucose. So no insulin, glycogen being converted into glucose. Glucose released from the liver. Didn't put that. Less food in the gut. Yeah, less glucose in the gut. Fine, but it's only 2 marks, so we got that. Right, both are heterozygous. So the answer is A. Yeah, that's correct. Punnett square. Yes. This was the write-off. Random roll of choice. 2 marks. So you get a mark for random and you get a mark for fertilization. Oh, beauty. I was thinking I was going to lose a mark there. Right, Corms. So you'll see that the mark scheme sets it out exactly how I've written it. Right, if you set it out like this, this is how the mark scheme is set out. So set up 2 boxes, interconnected, same biomass of decomposing apples and bananas. So yeah, rotting apples and bananas. Same species of fly, age or sex of fly, yes. Repeat, so yes, we've got that mark. Record the number of flies, yes. After a stated period of times, we have to give an example, so I did, that's an hour. Controls, age, number, mass of fruit. Biomass of the apples and the age of the fruit, yeah. But biomass of apples and bananas, that counts. And temperature, distance from the fruit, site container, release the same number of flies. So 100 flies, I got that as well. So easy, easy 6 marks there. Right, nearly done. 8. Can we get 100%, people? Maybe. Right, B, C, C. Yes, yes, yes. Palisade mesophyll cells near the top. Yes. To absorb as much light as possible. Yeah. So many chloroplasts, or lots of chloroplasts. Oh, I said chlorophyll. Oh, yeah, lots of chlorophyll to absorb as much light. Yes. They're densely packed together. Yes. Spongy mesophyll cells near the bottom for absorbing carbon dioxide. Yes. Less densely packed for diffusion. Yes. Good. Right. Easy 4 marks. This one I'm going to assume is correct because I've done the right thing. So I've divided by 100. So it wanted 2.9 centimeters or 29 millimeters. But yeah, so I'm going to say that I would have got that correct because we'd measure it with a ruler, which I couldn't do on here. Right, hydrophytes. Lower surface in the water, stomata would be in the water. Yeah. Upper surface is exposed to air where we can absorb carbon dioxide. Yes. Oh, that's 2 marks. There you go. Nice and easy. Allows transpiration. Yeah, allows transpiration. There we go. Right, B cells. So antibody, vague mark scheme here. Okay, have I mentioned antibodies? Yes. That they're specific. Don't like that, but okay, they bind to a specific antigen, yeah. They have I mentioned antigens? Oh, you get a 2nd mark for antigens. Okay. They kill, destroy pathogens, make memory cells. We mentioned memory cells there. Okay, good. So 3 marks. Phagocytes ingest bacteria, right? So what did it want here? Killed, broken down by enzymes. Broken down by enzymes. Products absorbed. Easy. 3 marks. Concentration of vitamins. Oh, hold on, missed one. A, correct answer. Concentration, yes. Oh, we're getting there for 100%, you know. Concentration of phagocytes. Yeah, concentration. Species of bacteria, yes. Vitamin C increases ingestion by phagocytes, yes. Or engulfment is the word for that. Only done once, not repeated. Yes. Not done at 37, yes. Not done in vivo. Done in vitro. Okay, that's the same as not in vivo. Bacteria, not only pathogens. Not all pathogens are bacteria. Oh, lovely. Final question. Right, we're getting on for 100. I don't want to chance it, but right, stigma style ovary, yes. You. Can't quite see. Oh yeah, okay, okay, that's fine. 2 marks. Going down the style, entering the micropyle. Yeah. Ovule becomes a seed, ovary becomes a fruit. Right, classic. Oh, you get a mark for the seed coat. So the ovule becomes seed coat. Tester. Okay. Right, percentage change, which I nearly messed up, I think. 10 point, yeah, okay. So I nearly messed up on this. So I could have cost myself 2 marks for that just from being a bit stupid and not reading the question properly. But again, when you get a dodgy result on your calculator, don't just assume that's correct, right? Just double check yourself, which is what I did. Because it said percentage increase and I got a decrease. So I've clearly done something wrong in my calculator. Suggest why fewer seeds germinate. This was interesting. In wet conditions, there are more bacteria. Ooh, I don't like that. Have I lost a mark? So seeds are diseased. Ooh, no. Enzymes are activated in storage. And you have to say in storage by the look of it. Oh, I've lost a mark. Seeds have already germinated in storage. Right. So already germinated, I get 1 mark. I did say that enzymes are activated as well, but I had to say in storage. Oh, -1. That's right near the end. Radical is seen, or the plumule is seen. Yeah, good, nice. Oh, guys, I lost a mark. Oh well. Right, hopefully you found that useful. I'm not going to blabber on too much more because that took me long enough. Right. Hopefully that's useful to you. I'm just going to stop the stop sharing. Hopefully that is useful to you to see. Again, I've still got 15 minutes left and I've marked the thing. So that's how you should be approaching the paper. You should have maybe not quite half an hour at the end, but you should certainly have 15 minutes, really, I'd be thinking. You should have at the end to check through your answers. Check units, check your conversions, check to make sure you've given data in explain questions when you're explaining graphs and tables. Make sure you've not gone through the origin on your graph unless you feasibly can. Make sure your definitions are correct. Make sure you're getting good terminology in there, and all will be well.